Okay, so allow me to share my PowerPoint that I will be discussing. That is all about the tools for studying nucleic acids. So this topic was given to me by our very own instructor. So I am Card Oliver C. Ricardo, MST. So when we talk about tools for studying nucleic acids, these are actually essential for understanding the structure and function of a DNA and even RNA when it comes to biological processes. So these techniques actually enables us to analyze the genetic material, explore gene expression, and also investigate the genetic variations, providing a more valuable insights into genetic, molecular biology, and biotechnology. Okay, so here are the topics or contents on my topics. First, we have the gel electrophoresis, followed by the PCR, or what we call the polymerase chain reaction. Then we also have restriction and modification. We also have here the gene cloning and gene sequencing with dideoxynucleotides. And lastly, we have the southern blotting. So all of these actually have their own connections and also differences. So all these techniques are actually interconnected when it comes to the molecular biology and also genetic research. So as they often work together, so in various applications, like for example po in identifying or in manipulating genes. Okay, so for example, PCR is frequently used to amplify DNA for analysis via gel electrophoresis. And kapag naman po gagamitin natin yung cloning, so it also involves here the restriction of enzymes and at the same time yung pagsisequencing po natin to confirm po if the DNA is inserted or hindi po. And para naman po sa ating southern blotting, it can actually, so we can actually utilize the use of, uh, utilize to amplify or to clone the DNA to detect and visualize specific sequences in complex samples. And for the differences naman po ng itong anim na to, so pag gagamit po tayo ng gel electrophoresis is para po paghiwalayin natin yung mga DNA fragments based po sa kanilang laki. Okay, so kung DAX ka, ibig sabihin mas mahirap kang gumalaw. So nasa huli ka. And then kapag naman Juts ka, charot, mas mabilis kang gumalaw. So ikaw yung nasa harapan. Okay? So while PCR naman po ay nagpaparami ng for a certain um, DNA sequences and so on and so forth. So, okay, so let's move now with the introduction to nucleic acids before we dive into the different tools to study nucleic acids. So let us define nucleic acids as a large biomolecules that actually stores and transmits genetic information, making them actually vital for life processes. So they are actually responsible for passing the heredity traits and instructions from one generation to the next generation. And then we have here the two main types of nucleic acids. So we have the DNA or what we call the deoxyribonucleic acids and at the same time the ribonucleic acids or the RNA. So kapag sinabi mo natin DNA again, refresh lang po natin. So DNA actually contains the genetic blueprint for an organism's development and also for function. While RNA naman po, it plays a key role in converting the genetic code in DNA into proteins. And the role of nucleic acids in cells is that they are actually essential for heredity, coding, protein, and protein synthesis. Okay, so we have here nucleotides. So we know that the building blocks of nucleic acids is actually nucleotides, okay? So, it actually consists of a sugar. So, yung sugar natin dito is either deoxyribose or a ribose sugar. And then we also have here a phosphate and at the same time the nitrogen base. So, for the structure of our DNA and RNA, so kapag sa DNA, we have the double helix structures that was discussed during the introduction po ng ating topic. And then, we also have here the RNA as a single stranded. So we have here the 4 nitrogenous base for DNA and for the RNA. So pagdating ng DNA, so we have here the adenine, cytosine, guanine, and thymine. Actually, halos same lang naman sila actual ng DNA for the RNA. Yun nga lang, when it comes to RNA, yung thymine po natin sa DNA is that mapapalitan na po siya ng uracil. Okay? So, this is actually the structure po ng ating DNA. And then, balikan lang po natin yung central dogma. Yung, kung ano po yung nangyayari during the central dogma that was discussed last meeting by Mon. So, for the central dogma, meron pa tayong tatlong steps. So, we have the transcription, translation, and also the replication. So, for the transcription method, so DNA is actually converted into the RNA acting as a messenger po. And then when it comes to translation naman po, so nagde-decode siya, okay, to form a specific proteins in the cells. While in replication naman po, so DNA copies itself to pass the genetic information during the division and then during the replication using the DNA polymerase to synthesize a new DNA. Okay? So I have here po a few questions. So first question is that what is the process by which DNA is copied into mRNA? So the answer is transcription. Okay? And then another question po, so during the DNA replication, so which enzymes nucleotides to the growing DNA strand? Again, ito yung pag-form po ng bagong DNA in the process of replication, that is DNA polymerase po. Okay, so the correct answer is letter C, DNA polymerase. And then, so true or false question naman po, so DNA is transcribed into RNA in the nucleus and translated into protein in the cytoplasm. Is it true or false? Okay, so the correct answer here is true. Sayang, magtatawag sana ako ngayon ng magre-resign. So, let's have here the overview po of our RNA. Again, RNA structure, it's a single-stranded and more flexible than the DNA. And then we have here the types of RNA. We have the mRNA or the messenger RNA. We also have the tRNA or the transfer RNA. And then meron din po tayong tinatawag na rRNA or the ribosomal RNA. So kapag dating po then kapag sinabi po natin mRNA, so it actually carries the genetic code from DNA to the ribosomes where proteins are actually synthesized. Okay? And then for tRNA naman po, it actually transports yung tipo natin, it serves as a transportation po for specific amino acids to the ribosomes during po the protein synthesis. Okay, and then for the rRNA, so it's actually a chemical component that facilitates yung assembly po or pag-assemble ng amino acids natin into proteins and then para din po ma-ensure yung proper translation ng ating mRNA. Okay? So this is actually the structure of our RNA. So for RNA, we have here the 4 nitrogenous bases. We have the cytosine, guanine, adenine, and uracil. Okay. So let's proceed. So I have here a question again. So which nitrogenous base is found in RNA but not in DNA? So the answer here is uracil. Ayan. So another question for RNA. So during the DNA replication, so which enzyme adds nucleotides to the growing DNA strand? Is it mRNA? Is it tRNA? Is it rRNA or snRNA? Of course, no, hindi na automatic kasali dyan yung sRNA kasi we only have three types of rRNAs. Okay, so the correct answer here is letter B. We have the 3tRNA. Okay, and then true or false? Ayan. So DNA is transcribed into RNA in the nucleus and translated into the protein in the cytoplasm. I think bumalik ata yung question or not. So the correct answer here is true. Okay? So ano ba ngayon yung importance of nucleic acids? Bakit merong nucleic acid sa ating katawan? Okay, again, katulad nga ng sinabi natin kanina, it's actually the genetic makeup of an organism. Okay? So we have here first genetic information storage. So understanding DNA is essential for tracing the inheritance, yung patterns, and also yung pag-study ng mga genetic diseases. As it provides insights to into how traits are passed from one generation to the next generation. So yung knowledge na ngayon nito na nagagather natin, it actually helps us to identify more kung ano yung mga other genetic disorders na meron. Kasi hindi lang naman po iisa, hindi lang naman po dalawa yung mga genetic disorders na meron. Okay, there are actually more than hundreds or thousands of, hundreds maybe, ng mga disorders po. So let's proceed with the next importance of nucleic acids. We have the protein synthesis. So for protein synthesis, so it's actually crucial role in translating the genetic codes into the functional proteins by carrying the instructions from the DNA and also yung RNA. So RNA now ensures that the correct amino acids are assembled in the proper sequence to form necessary proteins. Okay, and then we also have here the biotechnology. So studying nucleic acids is critical for the advancements in genetic engineering, forensics, and personalized medicine. Okay, so any questions po? Ayan, so I think none. So let's proceed now with the first tool to study nucleic acids. So the first one, we have the gel electrophoresis. So gel electrophoresis by definition is that this is actually a laboratory technique that is used to separate nucleic acids such as DNA and RNA or proteins based po sa kanilang size. Yun din po yung sinabi ko kanina na ang gel electrophoresis po is naka-base dun sa size, kung ducts or jutes. Okay? by applying po an electrical field across a gel matrix. So, yung method na po na to is actually allows us to analyze and compare the biomolecules more effective. Okay, so ano ba yung process during the gel electrophoresis? So in gel electrophoresis, DNA, RNA, or protein samples are actually loaded into cells or into wells with a gel matrix. So typically po, it is actually yung wells po natin are made up of agarose and polyacrylamide. Okay, so later on, i-discuss po natin kung ano po yung composition ng agarose natin and also yung ating polyacrylamide. So when an electric current now is applied, so the molecules now starts to migrate through the gel with they're moving, being influenced by their size and charge. So for example, ganito po. So of course, when dealing with let's say DNA, so magkakaiba po yung size nila. So paano natin sila ngayon i-identify? How are we able to arrange this thing? So the principle behind po of diesel electrophoresis is that it actually relies on the fact that small smaller molecules move faster and travel farther through the gel than the larger one. So as a result po, the samples now are sorted based on their size, and later on, it will enable us to visualize and analyze now kung ano yung distinct na bond present dun sa mismong sample. Okay, in the gel after the electrophoresis is complete. Take note that DNA and RNA are negatively charged molecules. So as they will be pulled later on towards the positively charged end of the gel. Okay? So the importance of gel electrophoresis in terms of molecular biology is that first, for DNA fingerprinting, so it is actually used in forensic science to compare DNA samples to identify, matching, and criminal investigations. So yun po yung ginagamit nila. Okay? And then we also have here for genetic research, it facilitates the analysis of genes, gene expression, and mutation, which helps understand genetic diseases. Okay? So for diagnostics naman, so it's basically used in medical for to detect the genetic disorders, yung infections, or even cancer makers through analysis of DNA or protein samples. Okay, so it is actually more evident or mass nature when it comes to diagnosis of certain diseases. Okay, so we have here the photo of our gel electrophoresis. Now that we have a bit knowledge, breakthroughs when it comes to molecular biology in the 20th century. So etong equipment na to is what we call now the gel electrophoresis. So which is actually a method of separating yung large molecules like segments of a DNA. Okay, so we have here, so when we introduce some concepts in biotechnology, we can actually, or we talk about how we can make a recombinant DNA plasmids. So yung DNA plasmids natin na yun can be inserted to a certain bacteria to produce many copies of gene or many copies of the protein that is produced when a gene is actually expressed. So, for example po, na gusto nating, we wanted to cut a plasmid and analyze kung ano yung, and and we wanted to make sure na etong bagay na to is copied as desired. Kasi kapag hindi yun yung desired na kakayain natin, baka mas lalo magkaroon pa ng disorder yung patient. So we need to make sure na specific yung ikakopy natin na DNA. Okay? So for example, we have here a mixture of DNA fragments. Of course, kapag sinabi natin mixture of DNA fragments, papasok na dito yung mga different sizes, different structures ng ating DNA fragments. Or, yeah, so let's try to imagine in a certain classroom na sobrang gulo. Yung iba, yeah, or before mag-start ang flag ceremony, syempre hindi naman pwede na yung mga matatangkad na sa harap or masyadong magulo yung arrangement nila. So anong kailangan natin gawin? So kailangan natin silang i-arrange. So the same is true with the gel electrophoresis na ganun yung gagawin natin. Okay, so we have here a mixture of DNA fragments that we wanted to separate and visualize. So With the help of gel electrophoresis, this actually enables us to visualize and analyze, and it works as follows. Ayan. So, in this tray, kung makikita pa natin tong tray, we have, there sits a slab of either a polyacrylamide or agarose. Ayan na po yung sinasabi ko kanina, yung composition ng ating wells. So, kapag sinabi nating agarose po, this is a most commonly used to run electrophoresis. Why? Kasi it is more simple to prepare and handle this in comparison with the polyacrylamide. So ano ba yung composition nitong agarose? So it is actually made from a polysaccharide that is extracted from a seaweed. So forming a more porous matrix that allows po yung mga larger molecules like DNA to move through. Okay, in comparison with the polyacrylamide, although yung kahit pa mas mahirap gamitin yung ating polyacrylamide, but the resolution when it comes to the polyacrylamide acrylamide is higher than that of the agarose. So this actually provides a more finer separation of the smaller molecules, especially dealing with DNA fragments and proteins due to its tighter gel matrix. Yun nga lang, mas complex yung preparation when it comes to polyacrylamide. And also, it requires a careful handling and preparation of this synthetic polymer. Okay? So this is actually the structure or image for agarose and polyacrylamide. So, paano ba tayo mamimili ng eto? Can we use agarose in that sense, or paano natin sila gagamitin? So, paano tayo mamimili ng specific na kung ano pwede natin gamitin na gel type? Okay, so first and foremost, dun sa molecule size. Take note that kapag gagamit tayo ng agarose, it's actually more for the larger DNA molecules. While in polyacrylamide naman po is smaller po yung fragments nila. Okay? And then when it comes to application of specificity, so for agarose gels, we actually prefer them to it's actually more of a routine use or DNA analysis. When it comes to polyacrylamide gels, so precise yung kanyang separations or yung pagsisequencing ng mga proteins. And then lastly, for when it comes to practical considerations, agarose gels are much more easier to prepare and handle. While polyacrylamide naman po requires more technical expertise. Okay? So we have here parameters of comparison for both agarose and polyacrylamide. So for toxicity, agarose is not toxic, while polyacrylamide is slightly toxic. When it comes to pore size, larger yung pores ng ating agarose, kaya mas okay siyang gamitin for a larger DNA. And then while polyacrylamide is smaller yung pores niya, kaya it's much more better if yung samples natin are in a smaller form. And then uses of agarose is mostly used to separate DNA and RNA, while polyacrylamide is used to separate protein and to also quantify it. And then for zero poring position, yung pagpapore pa natin for agarose is horizontal position, while in polyacrylamide, that is in a vertical position. Meaning po pa tayo. Okay, yung sa ating polyacrylamide. And then dye, yung dye pa na ginagamit natin for agarose is ethidium bromide, while polyacrylamide naman po is we have bromophenol blue. And then, sa pagre-reuse po natin, agarose can be melted and be reused. So much more sustainable, I guess. While in polyacrylamide, it's not normally reused. Okay. So, let's move na po. So, for example, meron na po tayong sample. So ito na po yung mga maliliit natin dito. Ito na po. Ito na po yung sample natin, for example. So yung sample sa natin is ipo-pour na natin ngayon sa ating gel electrophoresis. So at the one end po, there are a series of wells. And a number of samples can be loaded dun sa wells. Kaya kung mapapansin nyo dun sa activity 4 natin, using digital electrophoresis, so we have po different lanes. So yung iba, may 4 lanes, and then may 7 lanes, and so on and so forth. Okay, so each of these actually is a mixture of some DNA molecules of varying length. So this apparatus is also equipped with electrodes at each end. And those are the cathode and our anode. Again, with cathode po, they are the negatively charged electrode, while anode naman po, they are the positively charged electrode. Okay, so once everything is loaded na po, and ready na po siya mag-go, i-open na lang po natin yung power source. Okay, and then, if we're going to recall, yung mga phosphate group lines natin when it comes to the DNA backbone, and that each phosphate group, meron po silang oxy anion. Okay, yung oxy anion po na yun carries now a formal negative charge. Kaya yung sinabi ko kanina, yung sa parang introduction is that DNA and RNA are negatively charged electrodes. Okay, they are negatively charged. Okay, so this means that the negatively charged cathode will repel the DNA molecules and they will begin to travel along the gel towards the positively charged anodes to which they are more attracted. Take note, pag negative and positive, mas malakas po yung attraction. So imagine your partner being a more positive and you being negative. So mas malakas ngayon yung attraction ng dalawa. Charot. Okay, so personal experience. Amen. Okay. So the thing is that this gel is actually sticky porous substance and the molecules have to migrate through the pores to move along the gel in a process called sieving. So kapag sa agarose, mas malalaki po yung bilog-bilog or yung mga pores nila. Imagine, ano kayong sa akin? Malalaki kayong pores or maliliit yung pores. Charot. Is it more of agarose or polyacrylamide? Charot. Okay, so the larger the DNA, the more difficulty it will have in navigating through the pores. Okay, kapag mas malaki, mahirap talagang papasok. Diba? Imagine, charot. Okay, so which means that smaller DNA molecules will travel greater distances through the gel, while naman po yung mga larger ones will travel shorter distance through the gel in the same time travel. So yun na po yung sinasabi ko kanina na kapag mas malaki yung size ng ating DNA fragment, that mas mahirapan siya mag-move in comparison with yung mga maliliit natin na mga DNA fragments or DNA strands. Okay? So yung process na ito ngayon is much more reliable and quantifiable that we can actually plot and approximate the number of base pairs in a DNA molecules as a function of the precise distance as it travels during the gel electrophoresis. Okay? So once once yung separation na natin is complete, the current is turned off. And a DNA binding dye is added now into the system that glows a fluorescent pink in UV light. Like for example po, ito, ganito po yung mangyayari, parang iilaw po siya. Okay, so this is how the data is gathered, which will show up as thin bands and that sometimes resemble as a ladder. So if many different DNA molecules were present in the sample and each band, then it contains thousands of identical DNA molecules of that particular length. Like for example po, ito. So itong specific po natin dito is marami po siyang kagaya. So, ayan. So identical siya with other DNA fragments. So farther, and then yung last thing naman po natin, farther yung distance niya from the well So meaning shorter yung kanilang DNA molecule. So in comparison with this one, na shorter yung kanyang distance from the well, so meaning larger yung ating DNA molecule. Okay? So I hope you understand po. Okay, so again, ganito po yung nangyari, pero charot lang to kasi GIS lang. So as I said po, so these techniques may be used to chop up the plasmids and with restriction enzymes and analyze the results. So it can also be used to assess the products of gene amplification using the PCR. Diba? Yung sinabi ko kanina, habang while using the PCR method, pwede natin, ginagamit natin yung gel electrophoresis. Okay? So, what now are the factors affecting the gel electrophoresis? Kaya, the first one is the gel concentration. So, higher gel concentration for smaller fragments, while lower naman po yung concentration when it comes to larger fragments. Okay? So, for voltage applied, so higher voltage or applying a higher voltage during the electrophoresis increases the increases the migration speed of the molecules through the gel, allowing na mas mabilis po yung kanilang pag-separate. Okay? So kapag mataas yung energy, mas mabilis maghiwalay. Sad. Sarot. And then lastly po, for buffer solution, it maintains pH and conducts the electric current for a consistent separation. Okay? So what now are the applications of gel electrophoresis when it comes to DNA analysis? So the first one is that when it comes to DNA fingerprinting, so DNA fingerprinting compares the DNA samples to identify individuals useful in forensics, and also when it comes to paternity tests, and lastly, yung personal identification. Take note na yung fingerprints natin is very unique sa lahat ng tao. And then, another one is that PCR product analysis. So, verifies the size of the amplified DNA fragments after the PCR. Kasi yung gel electrophoresis natin is based on the sizes ng ating mga DNA fragments or DNA. And then we also have here the DNA sequencing, so which is used to separate, or DNA sequencing uses gel electrophoresis to separate DNA fragments, so helping na yung pagdetermine na ngayon ng order ng ating nucleotides in a DNA molecule. And then we also have here the genetic mutation detection, so which detects mutations by comparing the altered and normal DNA sequences. Again, kapag mutation, merong disorder na dun sa gene or DNA. And then lastly, we have here the restriction fragment length, or the RFLP. So that analyzes genetic variations by fragmenting DNA using a restriction enzyme. Okay, so do you have any questions po? Ayan. So if none, I have here a few questions din po when it about the gel electrophoresis. The first question is, what is the main purpose of gel electrophoresis in DNA analysis? Okay, so the main purpose po of gel electrophoresis in DNA analysis is actually to separate DNA fragments by size. Okay, another question is that what determines the speed at which the DNA fragments move through a gel concentration? The correct answer is the fragment size. Di ba kapag mas maliit, mas mabilis yung move. Pag mas malaki yung DNA fragments, mas mabagal yung movement nila, yung speed. Okay? True or false? DNA has a positive charge causing it to migrate towards the negative electrode. False. Again, DNA and RNA is a negative charge. Okay? Ayan. So, what is the name of the dye used to visualize the DNA in an agarose cells? If you can still remember yung kaninang table natin. So, we have the ethydiubromide. Okay, very good. And then we also have another question. So what does the thickness of a DNA bond on the gel represent? Ano kaya? It is the concentration of the DNA. Okay. Last question po for gel electrophoresis. So the distance of a DNA fragment travels is inversely related to its size. True or false? Is it inversely or directly proportional? The correct answer is true. Ayan. So, do you have any questions? Okay, so if none, we will move now to the next tool to study nucleic acids. Okay, so let's continue. Next topic is polymerase chain reaction, or we have the PCR. Okay, so we identify PCR or ano ba yung technique under the PCR. Okay, so PCR is actually used, or it's actually powerful, okay, technique that is used to amplify a specific DNA sequences that is used to generate um, generate millions of copies quickly and efficiently for an analysis. Okay, ano ba yung key enzymes na ginagamit during the PCR? So it utilizes DNA polymerase to replicate targeted DNA regions exponentially in a short period of time. So the temperature recycling, so this actually involves a temperature that is being repeated through the process of heating and cooling steps that is used to denature, anneal, and also extend the DNA. So we also have here for the sensitivity, so they are susceptible, allowing the detection and amplification of even trace amounts of a DNA fragments. Okay, so history of PCR. So it's 1983, Dr. Kari Mullis developed the PCR, and in 1988, so Parkin Elmer introduces the automated thermal recycler or cycler. And in 1990s, amplification and detection of specific DNA sequences using the fluorescent DNA binding dye, laying the foundation for future real-time or kinetic PCR. And then in 1981, so we have here the RT-PCR that is developed using a single thermostable polymerase or RTTH. So facilitating diagnostic tests for RNA viruses. So actually, RT-PCR natin, hindi lang nagamit yung term na yun during the pandemic. But meron na po talagang RT-PCR during that time. And in 1999 po, we have here the Dynal launches or the DRB36 HLA typing kit for a tissue typing. And then in 2003, we have here the HIV-1 monitor test version 1.5 product. Okay, so this is actually the machine na nangyayari na ginagamit for the PCR. Okay? So, let's discuss one by one yung mga topics under the PCR or yung mismong process. Okay? So, have you seen a photocopier machine? Like this one po. So, of course, lahat naman tayo is suki nung college. Okay? Sa paggamit ng mga sa pagpapapotocopy ng mga modules, ng mga activities na pinapauwi ng mga teachers, and then mga reading assignments, and so on and so forth. So, when it comes to biotechnology naman, meron tayong tinatawag na polymerase chain reaction that is used for the DNA. Okay? So, polymerase chain reaction or PCR provides a way to make more copies of a DNA portion and lots of DNA copies. And this technology does not happen in a cell either. So nangyayari siya sa test tube. Again, is it in vitro or in vivo process? You can still remember. Okay, so in fact po, yung ating PCR, it actually happens in a test tube. Okay, so paano nga ba let's try to answer these two questions. But let's focus first on how does PCR work. Okay, so when we need to, what we need, ano ba yung kailangan natin before po magkakaroon muna ng PCR analysis. Okay. So, of course, we need supplies. Ano ba yung mga supplies na kailangan natin kapag mag-undergo ng PCR? The first one is that we have the DNA portion that we want to make a copy of. And also, we need a buffer to also put it in. And then, we also need things that would be necessary to make more copies of DNA, such as primers. Okay, so recall that primers actually help DNA polymerase, which is a building enzyme, know where to go start its building, kung saan ba siya magsisimula. And then we also need a DNA polymerase. So in DNA polymerase, that's the building enzyme. And then take note that a DNA polymerase is often used as a heat-resistant type of a DNA polymerase, as PCR requires heat. Okay, and then we call that as a Taq polymerase. So, which is a type of heat-resistant DNA polymerase. So, if you can take that heat, get out of the hydrothermal vent. But, luckily, meron tayong tinatawag na polymerase, Taq polymerase. Okay, so Taq polymerase is originally from a type of a bacteria that can handle and live in really high temperatures of nature, especially in hot or springs. Okay, so we also need DNA nucleotides for the DNA polymerase to build with. Okay, so right after po ng ingredients natin, kunwari lang magluluto tayo. So meron din po tayong sequence or what we call the PCR sequence. So sa PCR sequence, we have here 3 major stems. So we'll illustrate with one double-stranded DNA molecule here, and let's assume that yung bagay na yun is yun yung kailangan nating i-replicate or yun yung gusto nating i-replicate. So the first step in the PCR sequence is that we have the denaturation. So during the denaturation, the naturating enzymes was used. So we need heat actually. So when the naturating enzymes, we actually need heat. Okay, and heat is what we will use in this step. Okay, so ano bang nangyayari dito? So this step actually involves the addition of heat needed to separate the 2 strands of the DNA molecule. And then, after po, magkakaroon na po ng separate na. So yung DNA natin, maghihiwalay na po sila. Okay, with the process of denaturation, which involves heating. And then, we also have here annealing. So kung mapapansin mo natin dito sa annealing natin, mayroong mga snowflakes. It means to say na malamig po yung part dito. So the 2 DNA strands that have been separated by that of our denaturation are going to be cooled and be joined by the primers. So kung mapapansin po natin yung primers natin dito, nag-bind na siya dun sa end ng ating separated na DNA. So the temperature for this step should allow the primers to bind to a specific segment of the DNA that we wanted to amplify, which actually means that gagawa na tayo ng mga copies nito. Okay? And then last, for the DNA synthesis, take note when we um, take note that the definition of synthesis is to make something. Okay, so with the DNA synthesis, we're going to make copies of the DNA. So magpo-proceed na po siya. And to do so, DNA polymerase will begin to work on both of these strands, and it will use the DNA nucleotides as its building material to amplify the DNA. So we should note that the temperature at this step may be a little warmer than the previous one. Kasi nga, cooling lang po yung nangyayari during the annealing. Okay? So it's step as it needs to be a temperature that is actually ideal for a specific DNA polymerase that is being used. Okay? So, yan. And then, so this is actually similar to, or how it would be in a DNA replication within a cell. Okay? So, kapag i-repeat po natin siya, again, mag-undergo po ng process na denaturation, annealing, and then the synthesis. So, for example, meron na tayo dito. We have 2 double-stranded DNA molecules to start with. And then, same process po, denaturation, annealing, and then DNA synthesis. And then, continuation naman po, kapag meron na po tayong 4 na double-stranded DNA molecules, and kapag i-repeat po natin yung step, so mag-undergo ng denaturation, maghahati sila, and then mag-undergo ng annealing, which is magbabind yung primers, and then synthesis na. Now, meron na pa tayong etops. And then kapag ulitin natin siya, ganun ulit po yung mangyayari. Again, it will undergo the denaturation, annealing, and then synthesis. So, ganun po yung mangyayari kapag times 2. So, madodouble po lahat sila kung ano yung ilalagay natin. So, this is what happens po. So, denaturation, yung temperature natin for that is we have the 94 degrees Celsius. While for annealing naman po, we have 54 degrees Celsius. And then for the extension or the synthesis, we have 72 degrees Celsius. Okay, so why do we need to use PCR? Para saan ba ang PCR? Well, any technology that needs copies a portion of a DNA could find PCR useful. Like for example po, when it comes to DNA fingerprinting in our gel electrophoresis, so we mentioned that DNA fingerprinting can be part of a crime scene investigation. Okay, so well, in order to have enough copies of DNA samples to run in a gel electrophoresis to analyze, PCR can be performed to make copies of the DNA fragments that are found in the crime scene. Kaya kung mapapansin po natin kapag mayroong crime scene, may mga kinukuha po sila using cotton buds or cotton po to get the sample. And then yun na po yung gagamitin nila to check kung kaninong DNA. To reveal kung sino ba yung salarin or killer maybe. Okay? So another example or kung saan pa natin gagamitin ng ating PCR is for diagnosis of disease such as caused by virus. Ito na yung tinatawag natin na COVID-19 or the SARS-CoV-2. Diba? Ano ba yung ginagamit natin? Paano natin masasabi na itong tao na to is infected by that virus? Diba? So magkakaroon ng virus testing. And then yung tinatawag na yun is what we have the RRT-PCR or the real-time reverse transcription or the PCR test. So, but to be more specific po, yung real-time reverse transcription is much more reliable or mas mabilis kasi siyang makikita natin yung results. Like, pagpunta natin for testing centers and then after 30 minutes, I guess, and then andyan na po yung results ng ating results kung COVID positive ka or negative. Ayan. So the reason for reverse transcription in there is because there is a viruses that uses RNA as its genetic material instead of using Gen A. And we have to use an enzyme called reverse transcriptase. phase that is used to convert RNA into DNA. So before we can do a regular PCR steps po that we have mentioned, ayan, we must convert po muna yung isolated and purified RNA into DNA. By how? By using the PCR. First, mag-undergo ulit siya ng denaturation, which actually separates the DNA strand. And then, mag-undergo na siya ng annealing. So, a specific primer now is added that will bind to and reverse the transcriptase, an area of that certain viral RNA. And then after which, it is used now to convert the viral RNA into the cDNA or yung tinatawag natin na complementary DNA. And then using specific primers in the top DNA polymerase, like what we've mentioned, so yung cCDNA na natin can be copied over and over, and then magkakaroon na po ng cycle, and then hanggang magiging familiar na po. Okay? But, kapag ganito, hindi agad natin siyang makikita. So the goal is that we need enough copies para sa viral cCDNA. In order for that to be more detectable, katulad dun sa gel electrophoresis natin kanina, gagamit tayo ng fluorescence. Okay? So yung fluorescent probes natin are actually used for identification. Identification to a certain level that is needed for a positive result. And if the virus genetic material is actually not present, then primers wouldn't bind, and therefore, hindi po magkakaroon ng complementary DNA. Okay? Wala pong cCDNA na copies ang maproproduce during that time. Kapag wala po. Okay? Kapag negative. Ayan. So ano bang purpose ngayon ng polymerase chain reaction? By the way, do you have any questions po? Ayan. So let's proceed po for the PCR. Yung purpose niya, first is for, again, DNA analysis, which amplifies small DNA amounts for research or testing. Then we also have here the disease detection. So PCR helps identify pathogens or any genetic mutations in clinical diagnostics, allowing for an early detection of diseases and informing treatment decisions. And then we also have here the forensic science, and then also the cloning genes, which is used to replicate specific DNA sequences for insertion into plasmids or vectors. Ayan. So we have here again few questions both regarding our PCR. So the first question is, which component of PCR binds to specific DNA sequences to initiate replication? Ano pa ba yung nilalagay natin dun sa part ng ating annealing? Para po para magbabind na po siya to a specific DNA sequences. So we use primers. Okay, so mapansin nyo ang daming dito, pero sa kanalang po yung ginto, meron na po yung sa DOST. Charot. Okay. So let's proceed. So which of the following is not a typical component of a PCR? A, DNA template. B, primers. C, DNA ligase. D, TAC polymers. Of course, if you can still remember yung ating ingredients kanina. Of course, DNA template is present, check. Primers are present in the process of annealing, for the process of annealing. And then we also have your TAC polymerase. And the component that is not part of the PCR reaction is we have the DNA ligase. Okay? And then a true or false question. So the annealing temperature is higher than the denaturation temperature in PCR. True or false? Is it higher? No. Kasi nga mas malamig dun sa ating annealing. in comparison with the denaturation. And then next question we have here. So what determines the specific DNA region that is amplified in the PCR? A, the amount of Taq polymerase ba? Ayan, so lumabas na yung sagot. Pasensya. Okay, so the correct answer here is the primer, that is B. So B, primary use. C, the amount of DNA template. D, the buffer solution. So kung mapapansin po natin, itong tatlo po yung A, C, and D, they are, dapat ang ano nila dyan is standard. So hindi po nagmamatter dun sa amount. Okay? So it's on the primer that we use po. Okay? And then another question is that the denaturation step in the PCR occurs at a temperatures typically above 90 degrees. And the melting temperature of the primers used during the annealing process depends on the melting temperature. Is it true or false? Of course, the correct answer here is true. Okay. So, any questions, Bob? So if none, we'll move now to the next topic, which is on the restriction and modification. Okay. So for restriction and modification, systems are actually crucial when it comes to defense mechanism that is employed by a bacteria and archaea to protect themselves from any foreign DNA. Okay, such as that introduced by bacteriophage, viruses that infect bacteria. So these systems consist of two primary components. We have the restriction enzyme and then at the same time the modification enzyme. So yung common bacteria dito is widely present in terms of prokaryotes, especially po sa bacteria for defense against bacteriophages or phage. Okay, so let's define what is all about the restriction enzyme. So restriction enzymes are also known as the restriction endonucleases. So recognize and cut specific sequences from invading organisms or from a foreign DNA. So by cleaving the DNA at the precise locations, that they prevent the replication of that unwanted genetic material. So para hindi na po magkakaroon ng replication at mas lalong magkaroon pa ng error or mutation, is hindi na po niya i-replicate. Okay? Doon na po papasok yung ating restriction enzyme. Kasi i-restrict niya na po yung growth ng unwanted DNA. Okay? And then we also have here the modification enzyme. So for modification enzymes, it's often methyl transferases. Okay, so these are actually methylate host DNA. So we need to add methyl groups to specific bases in the host own DNA. So yung modification na nangyayari dito is that pinoprotektahan niya na ngayon yung mismong host from being cut by the restriction enzyme and allowing the organisms to differentiate between its own genetic material and the foreign DNA. Sorry po. And then the purpose of restriction and modification systems, first is for defense against invaders. Okay, so restriction enzyme systems allow bacteria to recognize and cut a foreign DNA. Such as yung mga bacteriophages na tinutukoy ko kanina or yung mga plasmids using restriction enzymes that pinoprotektan na nila ngayon yung bacterial cell from a potential harmful or any invasive genetic material. And then we also have your facilitating genetic engineering. We also have self-protection. So for self-protection, ayan, so yung bacteria used for modification enzymes to methylate their own DNA at specific sites. Okay, so para po ma-prevent yung restriction enzymes from cutting the host DNA. And then we also have regulating gene expression. by cutting or modifying certain DNA regions. And then lastly po, we have your maintaining genetic integrity. Okay, so itong maintaining genetic integrity, sa part na to ngayon is that prevent niya yung mga unwanted changes na pagbabago in the bacterial genome by eliminating the foreign DNA that might cause any mutations. Okay? So for example po, this man, so inside po, sa loob ng ating katawan. So yung example ko na to is actually for genetic modification. So inside all our cells, like this kind of a cell, ayan. So all living organisms have a DNA. So this DNA actually contains instructions for a whole range of features that makes up an organism. So each segment now of a DNA has an individual set of instructions, which is called as our gene. Okay, so there are genes that determines the eye color, yung may nag-determine din kung straight ka ba or kulot when it comes to your color, and so on and so forth, and marami pa po. Okay, so for example po, instructions to specific cells in the pancreas that produces insulin, which is a protein that helps control our blood glucose levels. Okay, so there's actually a condition known as a diabetes. We all know that. Okay, so for diabetes, dito na yung part ng katawan na natin na hindi na natin nakokontrol yung blood glucose levels. So there are different types of diabetes, but basically it's a situation where the body can't either produce or can currently use insulin. Okay? So, ano na ngayon yung magiging solution natin? The very common solution is gagamit tayo ng regular injections of insulin, right? But, saan ba natin usually kinukuha ang insulin? That's actually the main problem. Right now, since advanced na po yung technology natin, gumagamit na po tayo ng human insulin instead of using pigs or even cows. Okay, so yung development tayo ng technology natin is very advanced, kumbaga. Okay, so we make human insulin in terms of injection of insulins. Okay, so we take the gene or yung genetic instruction for that human insulin and then insert that gene into the bacterial cells. So, and allow the bacterial cells to genetically modified or to make insulin for us. So, ganito po yung mismong nangyayari. So, bacterial cells have different circles of DNA in them, which we call as a plasmids or yung bacteriophages kanina. So ngayon, tatanggalin na ngayon natin yung plasmids and then use this and use a restriction enzyme to open them up. So ikakat po natin itong plasmids. Okay? So the plasmids now that we have is meron na siyang tinatawag na sticky ends. Okay? Yung red po natin doon, yun na po yung tinatawag natin na sticky end. So, now that we need the gene for the feature, ayan, that we want in this case, so the gene na for the human insulin from a human cell, we use the same restriction. Okay, gagamitin po natin yung the same restriction enzyme na ginamit po natin kanina sa pag-cut dun sa plasmids. And then with our plasmid to cut out the human insulin gene. So it now also has sticky ends. So ano na pong mangyayari ngayon? So the human insulin now, genes are then mixed with the open plasmids along with an enzyme called ligase. So yung ligase po natin helps join the sticky ends of the human gene to the sticky ends of the plasmid. So here, you can see what's known as the recombinant plasmid, which is a combination of the original bacteria DNA and the human insulin gene. Okay, so the recombinant plasmids now are then inserted into the bacterial cells, and then magkakaroon na po tayo ng recombinant bacteria. Okay, so the final step now is to grow the bacteria in ideal conditions in a fermentation. So itong tank na to allows them to provide and replicate many times all while producing po lots of human insulin. So based on the genetic instructions na kinuha natin doon sa human. Okay, so yung insulin na yun can be collected and purified and bottled ready for the human use. Ayan. So ganun po yung nangyayari natin sa ating restriction and modification. Okay, so question. So what enzyme cuts a foreign DNA at specific sites in the restriction modification system? Ito po yung ginamit natin na pag-cut, isang enzyme na ginamit natin na pag-cut for our plasmids and at the same time sa pag-cut dun sa human gene. We call that as our restriction enzyme. Very good. So another question, restriction enzymes cut DNA at the specific sequences which are often what? A, is it random? B, repeated? C, circular? D, palindromic? It is palindromic. Okay? So true or false, all bacteria have restriction modification systems for DNA protection. Meron nga ba? Lahat? All bacteria? Of course, false po. Ayan. So what process involves the protection of DNA by adding methyl groups? Ayan. So meron na po tayong keyword dun na methyl group. We call that as our DNA methylation. Okay. Another question. So what is the main purpose of restriction enzymes in a bacteria? A, is it for DNA replication? B, cleaving foreign DNA? C, protein synthesis ba? D, transcription? So the correct answer here is letter B, or the cleaving foreign DNA. Okay? So restriction modifications are systems only target bacterial DNA for cleavage. Take note, only targets a bacterial DNA for cleavage. Is it true or false? The correct answer here is false. So mali po. So do you have any questions? Okay, so let's continue our topic on the gene cloning. Okay, so for gene cloning, so the insertion of a DNA fragment of a DNA carrying a gene into a cloning vector. Yung vector po natin is a bacteria, okay? And a subsequent propagation of recombinant DNA molecules into many copies is known as a gene cloning. So it is actually used to create copies of a particular gene for downstream applications, like for example sa gene sequencing, sa mutagenesis, and then ginagamit po din po natin siya for genotyping or any heterologous expression of a certain protein. And then meron din po tayong ginagamit na traditional technique when it comes to gene cloning. And then ito po yung that actually involves the transfer of a DNA fragment of interest from one organism to a self-replicating genetic elements. Like for example, yung bacterial plasmids na ginamit din po natin kanina for restriction and modification. Okay, so ano ba yung purpose ng gene cloning? So the purpose of gene cloning is first, ginagamit po natin siya for the protein production. So it generates large quantities of specific proteins for industrial and therapeutic applications. So again po, kung babalikan po natin kanina yung nating discuss when it comes to restriction and modification po. So, it actually facilitates the generation po ng mga um, large quantities of a specific proteins through yung recombinant DNA natin. Okay? So, which is actually essential po for when it comes to men's or uh, health and also for industrial processes. Okay? So, we also have here the genetic modification, so which enables the genetic creation of genetically modified organisms with desired traits. Like for example, yung tomato. So at yung tomato is kinuha na ng specific gene para longer yung kanyang shelf life. And then yung ating golden rice, para yung nutrients na makuha natin dun is mas marami compared to yung natural na white rice. And then also, another purpose of gene cloning natin is we have the disease treatment. Okay, so for this statement, it produces therapeutic agents into the hormones and enzymes for medical use. So ito po is actually an essential for treating various medical conditions and improving the health of the patient. And then we also have here the process when it comes to gene cloning. So gene cloning process, again, it involves the use of bacteria to make a multiple copies of a gene. And then you also have here the insertion of foreign DNA. So foreign DNA is inserted into a plasmid, and the recombinant plasmid is inserted into a bacteria. So yung pag-insert na ngayon natin ng foreign DNA that contains the gene interest is inserted now to the plasmid. So which is a small circular piece of DNA that replicates independently within the bacteria. Okay, so once po yung foreign DNA natin is integrated doon sa mismong plasmid natin, the recombinant plasmid is now introduced into a bacterial cell through a nag-undergo siya ng process ng transformation. Okay? And then we also have here the bacterial reproduction. So when we talk about bacterial reproduction, so this is actually the recombinant plasmid, which is being introduced sa bacterial cell natin. Katulad nung kanina, so the bacteria is nagre-reproduce na siya, and then yung cloning na po ng plasmids natin along with the inserted gene. So magre-result na po siya ngayon ng production of a multiple copies of a single gene. Okay? So again, kapag sinabi nating clone, ito po yung exact copy ng isang organism. Like for example, ito clone, nanonood kayo ng Naruto or nung iba ng kabataan, nung bata pa pa, na matanda na pa. Sarap. Okay, doon sa Naruto, di ba merong Kagibunshi technique? I think, correct me if I'm wrong sa pronunciation. Basta yun, nagka-clone si Naruto. Yung cloning ni Naruto is just the same dun sa kay Naruto itself, yung the original na. So kung anong gagawin niya, ganun din yung gagawin ng iba. So ganun din po nangyari sa gene cloning natin. Yung gene po is just exactly the same with the original one. Okay? So yung gene cloning natin acts as in making copies of a single gene. So cloning can actually provide a pure sample of an individual gene separated from all the other genes that is normally shares with the other. Okay. So we have here, for example, yung cell extract containing plasmids that is isolated from a bacterial cell. So DNA cloning is a method that is used to isolate a particular sequence of DNA from complex to a simpler, or to any different DNA sequences. In order to clone a fragment of DNA, we need now to insert a vector. Okay, usually a highly modified fudge or plasmids that can actually replicate in the host. Okay. So generally po, yung plasmid natin is a vector that contains 3 elements. So a cloning site first is the cloning site where the foreign DNA can be inserted. Okay, and then a drug resistance gene that can destroy the antibiotics. And then lastly, yung ampicillin natin that allows selective growth of the host cell. Ayun siya. And a replication to allow the plasmid to replicate in the host cell to insert foreign DNA. Pero bago po mangyayari yun, we must first use a restriction enzyme to cleave the vector at the cloning site. Again, yung restriction enzyme is used to cut. Okay, para po mag-attach or para po magbabind, para po mag-cleave yung vector natin sa kanila. So here, for example po, we have here the eco R1, so which used to cleave the palindromic sequence. For example, we have here the GAATTC. Okay? To produce a single-stranded ends, meron na tayong, di ba nakat na? And then, katulad kanina, kapag nakat, yung dulo nun is sticky ends. Okay? And then this can hybridize with any piece of a DNA that has also been cut with the EcoR1. Okay, so by a foreign DNA containing the sequence. So in here po, we want to clone a specific or a digested EcoR1 and then mix with the cleave vector. And then, yung sticky ends na po natin of the foreign and the plasma DNA molecules are now hybridized. And it is sealed into a phosphodiester linkages. Okay, so ayan, magbabind na po siya sa ating DNA. So by the enzyme DNA ligase, creating a recombinant plasmid. Again, yung DNA ligase po natin nag-serve as parang siya yung nagiging base or pandikit natin dun sa plasmid. Okay? So, yun na po yung mangyayari. So, yung DNA fragment na natin is inserted now into the plasmid. So, it will look like this. Imagine na. Okay, so the fragment of an ampicillin resistance genes and a gene replication origin or a pool of a library or circular recombinant plasmids is created. Ayan. So each plasmids now carries a unique fragment of foreign DNA as indicated by this ito po, yung mga colored na ito na po yung mga recombinant. Yung mga colored na arrows. Okay? So next po, ang mangyayari na is that yung host cells natin, like for example, we have the E. coli bacteria cells. So doon na po mag-a-add. Okay, yung E. coli bacteria now is added with the DNA molecules. Pero before mag-add siya, kailangan po natin ng calcium chloride to make them permeable to the DNA molecules. And then, again, yung process na tawag na yun is what we call the transformations, wherein a few cells takes up the recombinant plasmid. So, while most other cells do not, sabihin, hindi na po siya magkakaroon. Okay? Pero yung most probably na meron, ganyan na po sila. So, ito na po yung tube natin, for example, containing the plasmids and an E. coli bacteria. So, kailangan po natin silang i-grow. So, yung pag-grow natin ng ito is we need actually a nutrient agar containing the ampicillin. Okay. Take note sa part na to is kailangan din natin na yung gagamitin natin is resistant siya to antibiotics. Okay, so the bacterial cells are poured into a plate of a nutrient agar containing the antibiotic ampicillin. With the ampicillin in the agar, cells now are resistant to the drug and can grow at 37 degrees Celsius in sa incubation, and then doon na po sila mag-multiply. Okay? So each cells now produce a separate colony of cells. Okay? So have you tried na po na sa antimicrobial property of, let's say, itatest natin yung effectiveness na to. Yung doorknob. Okay. And then, kuha tayo ng konting sample and then lalagay. Tapos kung mag-grow siya, sabihin. So parang ganun po yung nangyayari dito. So magre-replicate with the use of the nutrient factor. Ayan. So the non-transformed cells now lack the plasmid with ampicillin resistance. Yung gene na ngayon na hindi nag-transforms will die. Okay? Ganun po yung katulad kanina na kapag hindi siya nag-form, hindi po siya mag-form ng complementary DNA or yung cDNA natin kanina. Okay? So yung cells that have plasmids became resistant to the ampicillin will survive. Okay? So magta-transform siya and then magsusurvive na yung cells. So the replication origin allows now the plasma to replicate by using host cells enzymes. So yung plasma replication is independent of the host cells division. So but plasmids are distributed to each daughter cell. So yung replication na ngayon is independent on the bacterial chromosome replication. So when the host cells dies, ayan, so bacterial chromosomes replicates and then yung cells na ngayon is mag-divide. So at least one plasmid segregates to each daughter cells. Okay, so yan, so magbubuhay. And then yung cells na natin ngayon contains copies of the same recombinant plasmids with its fragment of foreign DNA will now have a variety of colonies or assays that can be used on the bacterial colonies para ma-determine natin yung isang part, that certain DNA sequence na gusto nating i-isolate. Okay, so ganun po yung nangyayari sa aning. Okay, so any questions? Okay, so let's proceed with the question, Q&A portion. So first question is that what is this small circular DNA molecule used as a vector in gene cloning? Ito yung paulit-ulit natin na sinasabi kanina. We call that as our plasmids. Okay? And then, another question. So what determines a specific DNA region that is amplified in the PCR? A, through sequencing the RNA. B, through DNA degradation. C, through antibiotic resistance. D, through cellular division. Ano kaya? So the answer here is letter C. So through antibiotic resistance. Okay? Kasi kapag hindi siya resistant, mamamatay siya. Hindi siya ma-amplify. Okay? And then DNA ligase is used to bind DNA fragments during gene cloning. Is it true or false? DNA ligase ba? The correct answer is no. Ayan. So what is the process of making identical copies of gene? Of course, we call that as our gene loop. Okay, another question. So which of the following is used as a host cells in gene cloning? Ano yung magiging host cells natin dito? Is it the bacteria? Is it the virus, fungi, or plants? Of course, X na agad yung plants. Okay? The correct answer here is the bacteria. So, transformation is the process of cutting DNA into fragments. Is it true or false? Transformation bang tawag doon? Correct answer is false. It's actually, di ba? Ang tawag natin doon is yung restriction enzyme. Yun yung way ng cutting natin ng DNA into fragments. Okay? So we will proceed now with our next topic on the gene sequencing with deoxynucleotide. So in here, ayan. So identifying genetic code. So gene sequencing reveals the precise order of nucleotides in a DNA strand, allowing now the researchers to identify a specific genes and understand yung mismong genetic information nila. So ano bang function ngayon ng deoxynucleotides in gene sequencing? So by determining the sequence of genes, actually pwede na nating makita yung kung anong functions and yung roles ng isang biological resources. Okay, and then advancing biotechnology, which is actually essential for developments in genetic engineering, diagnosis, and also for personalized medicine. Ayan. So, yung gene sequencing with dideoxynucleotides can be called as a Sanger sequencing. So, yung Sanger sequencing was actually developed by Frederick Sanger in 1977, which is a method for determining the nucleotide sequence of a DNA. So, yung method dito is that yung role na ngayon ng ating dideoxynucleotides are actually incorporated now into the DNA strand during the replication para po mag-further yung elongation natin. And then magkakaroon na po ng formation of fragments. So yung results na ngayon dito in the DNA fragments of different lengths, each with a terminating at a specific nucleotide, can now be analyzed and be sequenced. Okay? Resulting in a fragments of varying lengths. Ayan. So ano ba yung role ng ating dideoxynucleotide? So the first one is that when it comes to chain termination, so when a dideoxynucleotide is incorporated into a growing DNA strand, it locks the three or the position ng hydroxyl group natin sa sa third carbon. So okay, so effectively stopping further DNA is. So this actually now creates a DNA fragments of varying lengths. And then yung dulo niya na ngayon dun is andun na po yung dideoxynucleotide na pwede natin i-add. Okay? And then another one is that for fluorescent labeling. So each type of dideoxynucleotide, so we have here the adenine, thymine, guanine, and then cytosine is labeled with a distinct fluorescent dye for easy identification during the sequencing. Kaya kung mapapansin natin kanina, we go back to my previous slides po, yung arrows po nila is magkakaiba depending dun kung ano yung specific color ng isang thing. Okay? Yeah. So another one is when it comes to fragment generation. So as DNA polymerase adds or incorporates dun sa nalabelan natin kanina dideoxynucleotides or the modified nucleotides, so which is a mixture of a DNA fragments of different lengths that is produced, which can be separated for analysis or pwede natin silang i-deduce. Okay? Yeah. So, first, what is dideoxynucleotide? Ano bang dideoxynucleotide? So, Sanger sequencing is actually based on the dideoxynucleotides in the DNA polymerizing reaction. But, ano ba ang dideoxynucleotide? So, we all know that nucleotides is a composition of phosphate group, a sugar, and a nitrogenous base. So, yung nitrogenous base natin, we have can either be A, C, G, or C. Okay? So, for example, yung phosphate natin is absent, then it is called now as our nucleoside. Kapag nawala na po yung phosphate group natin kanina, matatawagin na natin siya, instead of nucleotide, magiging nucleoside na. And then if the hydroxyl group at the two end, atong sa baba po, with this one, if mare-remove po yung isa, then it is called as a deoxynucleotide. Okay? So the deoxynucleotides now that are present in the DNA are used as a substrate po by the enzyme DNA polymerase. Ayan. So yung all the DNA polymerase, whether it's it is used for DNA polymerase 1, 2, or 3 Taq polymerase or etc. Yung DDNP natin po is magagamit for their substrate. Kapag DDNP. Okay? So the role now ng hydroxyl group attached at the end of the third carbon and plays a very important role when it comes to DNA polymerizing reaction. So during this DNA polymerization, yung reaction po natin between the 3OH group and yung phosphate group natin of the NUCLEIC and TP is that kapag tatlo na po sila, if the 3OH or the hydroxyl group of the nucleotide is absent, kapag mawawala siya, then there would be no reaction. Wala po. Of course, kapag missing yung isa, walang reaction. Okay? So, hindi po magkakaroon ng addition ng new nucleotides and the reaction would stop. Okay? Hindi na po siya magkakaroon. Yeah. So, as 3OH group is absent, there won't be addition of new nucleotides in the polynucleotide chain during the polymerization po. So, the reaction would finally stop. So, thus, kapag yung nucleotide natin na nakatouch sa 3-hydroxyl group is absent na naman, tatawagin na natin sila as our dideoxynucleotide. Okay? Take note po na kapag sinabi natin di, that means 2. So, dalawang deoxy na yung nawala. So, dalawang hydroxyl group na yung nawala. Okay? Absence na ng dalawang hydroxyl group. Okay, so for example po, dito sa Sanger sequencing method. So, where the DNA or the template of the DNA is actually sequenced and let's say na-divide sa tat. So, each tube, so tube 1 to and 3 natin, meron siyang kanya-kanyang primer that will bind to the template of the DNA. So, yung all 4 DNTPs, like we have here now the D, yung N po natin dito is papalitan lang po natin ng A, T, G, or C. So, magiging DATP, VC, VT, VTTP, VTG, DTG, DCTP, okay, are present. Okay, so along with this, yung DNA polymerase natin po is also present. So yung reaction na natin ngayon will be the mixture and has a ratio labeled as a radio-labeled dideoxynucleotide at a very low concentration. So this means that by this tube, one will have the dideoxy ATP. Yung isa naman po will have the DDTTP. Yung isa naman po is DDGTP, and then the last tube will be DDCP. CTP. So ito po is nasa lower concentration. And all of them will be radio labeled. Itong apat po is ano po siya ng radio labeled. Okay, so let's see po kung ano po mangyayari kapag magkakaroon na po ng prime. Ano po yung unang magbabind? So in one tube, one DNA. So for example, we have here the tube 1, which is the DDATP. So in tube 1, that DNA polymerase will add the DNTPs on the template ng DNA and carry out the normal polymerization reaction. So, mag-attach na yung isa, and then mag-attach na po yung A, T, and then C, and then the other one will be attached. And then kapag babalik na po siya, so if somehow yung dideoxynucleotide natin is incorporated po, then the reaction suddenly stops. Kasi nga, bumalik na siya. Okay? So because the reaction terminates as there is no 3-hydroxyl group for the addition of new DNTPs, ayan. So yung incorporation na nga yun natin ng dideoxynucleotides and the termination of our polymerizing reaction is a random process. Sorry, nagbubungol na po. Ayan. So hence, yung overall reaction na po natin or yung mixture na po natin will have several fragments that has an experience of exchange termination. Okay? So now, if yung length na po natin ng fragment, matatawag na po natin sila as the location na yung ating adenine in the DNA and can be estimated kung gaano kalaki yung mismong fragments or yung DNA fragments. Kaya, if you can still remember, yung sa ating Zell electrophoresis po, parang ganito po, parang may mga ladders. So this is actually obtained by performing the polyacrylamide Zell natin using the Zell electrophoresis and the autoradiography. Okay? So yung X-ray film po natin, magiging ganito. So once the diagram is obtained, then ang makikita na po natin is yung sequence na ng ating information of the desired DNA fragment. So the first, we have the A. And then yung second po natin is again, magiging letter A pa rin po. And then the third one is that we have the letter G. And then yung fourth is we have the letter C. Ito po dito po natin siya naka A, T, G, C. Okay? And then what will be the fifth? It's letter T. How about the sixth one? Okay, again, it's letter C pa rin po. And then the next one, we have letter A, then followed by letter G, and then C, and then C. Okay, nakuha po. Yeah. So yung test tube na po natin, we have here the DDATP, tube 1, and then for tube 2, we have the DDTTP, and then tube 3, we have the DDGTP, and then for tube 4, we have DDCTP. So now in this method na po, each of these has a dideoxy nucleotide. Okay? So ito po have their own specific fluorescence lights. Okay? So each attached with a fluorescent light. So kung ano po yung ilalabas niyo na color, yun po yung present sa. Okay? So instead of mag-recreate ulit tayo ng panibago, ganito na po yung gagawin. So maglalabas na siya ng fluorescence. So it will now give a fluorescence when exposed to a UV light. So magkakaroon na siya ng kulay. Okay, so sana all makulay ang charis. Okay, so as each of the this dye, yung dideoxy nucleotide natin have their own fluorescence. And with that po, it is now possible po para ma-carry out natin yung reaction in one single tube rather than gagamit po tayo ng mga separate tubes. Okay, so mas mabilis na po natin siyang identify, and then it's much more easier now to identify kung ano yung mga andun yung present. So imagine kung isang tube lang it is much more easier. Unlike kapag yung ginawa natin kanina na 4 na tube. So mas lalong complicated siya. Okay, so the termination reaction now would result if yung dideoxynucleotide natin is incorporated in the DNA chain terminated fluorescence. Okay? So, ganito na po. So, the more advanced po yung systems natin na paggamit po ng capillary for this uncertain separation po, DNA fragments are further equipped with the CCD or the charge coupled device. So, ayun na po. So, magagamit na po natin to for the detection of a fluorescent sequence. Okay, so any questions po? Ayan, so let's proceed na po with another Q&A. So, don't worry po, last topic na po natin. May isa pa pala. Second to the last topic. Okay, so what type of labeling is used in Sanger sequencing to identify the nucleotide sequences? Ano pong ginagamit natin? If you can still remember po yung may kulay-kulay kanina, we call that as our fluorescent label. Okay? Another question is that, what component stops DNA elongation in Sanger sequencing? Diba? Ito po yung part na wala na pong reaction. Ano pong component no? Para mag-stop na po yung elongation curve. Para hindi na siya mag-produce pa. Para wala ng reaction. So, is it ribosomes? Is it dideoxynucleotides? Is it RNA polymerase? Or is it methyltransferase? Of course, the correct answer here is letter B. We have the dideoxynucleotides. Okay? Next question. So dideoxynucleotides are incorporated into the DNA strand to extend its length. Is it true or false? Mag-extend pa ba? The correct answer is false, di ba? Kasi nga i-stop niya na. So therefore, hindi na mag-extend yung length kasi nga makakat na doon. Okay? So another question. So what sequencing method uses dideoxynucleotides to terminate DNA strands elongation? It is the Sanger sequencing, or what we call as the gene sequencing with dideoxynucleotides. Okay? And then what method is used to separate DNA fragments in Sanger sequencing? Is it PCR, centrifugation? Is it gel electrophoresis, or the chromatography? I mean, is it? The correct answer is gel electrophoresis. Okay. Last item. So fluorescent dyes help identify the specific dideoxynucleotides in DNA sequencing. Is it true or false? Basically, no. The correct answer is true. Kasi ito na yung last part kanina na mas mapapadali na lang siya. So ito yung kahit gagamit na lang tayo ng isang tube to identify yung specific na base or fragment. Okay? So any questions po? So if none, let's proceed now with the next topic. Okay, so let's continue po my topic on the last part. Ito na po yung pinaka-last topic po. So which is all about the southern blotting. Okay, so southern blotting is actually a method used by scientists to find a specific DNA sequence in a sample that actually contains many different fragments. It's actually a laboratory technique. So ano pinangyayari dito? So first, it involves the transferring of DNA fragments from a gel onto a membrane where they are probed with labeled complementary sequence to visualize the targeted DNA. So it was actually developed by Edwin Southern in the 1975. So this method revolutionized the molecular biology by providing a reliable way to identify and study specific genes. But before po nagkaroon ng southern blotting, it was actually very difficult po to find a specific study or a study that focuses more on specific genes in a mix of many fragments. Okay, so it remains a cornerstone in genetic research, paving the way for the advancements in DNA analysis and for genetic diagnostics. Okay, so we have here the purpose. Alam, hindi ko po napalitan yung restriction and modification. So the purpose of southern blotting po, first is for gene detection. For gene detection po, again, it is used to identify specific genes in a complex DNA mixture. So that actually aids for diagnosis or even po yung genetic disorders. And then we also have here the genetic fingerprinting, which plays a crucial role or critical role for forensic science. Okay, and that helps to solve criminal cases and also paternity tests. And then lastly po, we have the genetic or the detection of genetic variants. So, yan po. So this is po, let's try to understand po kung ano pong nangyayari dito sa southern blotting natin. Okay, or southern blot. So the technique used for the detection of genomic DNA, so specific gene or DNA sequence and yung genomic genome po ang kukunin natin, or let's say ito po. So yun yung hahanapin natin. So for example, this one, itong specific area nito, yun po yung kukunin natin na specific DNA. So let's try to understand this technique in a more detailed um, in details when it comes to genomic DNA. So first, i-isolate po na muna natin yung specific gene na gusto nating i-kunin or i-identify. Okay, so we're going to use again a restriction enzyme para po i-cut. So fragments now generated by the restriction digestions are separated using the agarose gel electrophoresis. As there are many fragments po produced due to the restriction digestion. Ayan. So agarose gel is then treated with an alkali. Ito po yung mga NEOs. So these are actually based or what we call as the alkali. Okay, to separate yung double-stranded DNA natin in a single-stranded DNA in the DNA. So then, mangyayari po is that matatransfer na po yung siya to a membrane by placing the membrane po on top. Yung membrane natin na yun is what we call the nilo or nitrocellulose membrane. Okay, so by placing the membrane on the top of the transfer, so magkakaroon na po tayo ng capillary. Okay, so it usually occurs by capillary action once na-transfer na po natin silang lahat dun sa ating gel, sa ating agarose. Okay, so the membrane now is heated in a hot air oven at 80 degrees Celsius for 2 hours or exposed to a UV light. Okay, so this ensures that the DNA is permanently attached with the membrane now, or kung hindi man po, magkakaroon siya ng attachment din sa ibang fragments. On the next step, that actually involves the detection of the desired probe. And then, so yung fragment using a probe as actually a nucleotide, that can be used for sequencing the complementary to the desired DNA fragment which is to be detected by the probe. Actually, ito pong probe natin is tinatawag natin na PR2 or yung fluorescence dye. Okay? Ayan po. Which is watched po gamit po ang ating X-ray film which is placed on the membrane to perform autodiography. And then, magpapakita na po siya dito. Kung maalala nyo din po kanina yung autodiography, ayan, so the presence or absence po ng desired DNA fragment natin, lalabas na po. So the application na po of this southern blot, ayan, so is widely used po to detect kung present po ba or absent yung isang gene in a given sample. So suppose po, we have here the genomic DNA. So ito pong genomic DNA natin is andun po yung gusto nating i-identify. Okay? And then, for the restriction digestion, ayan, gagamit po tayo ng restriction enzyme para po i-cut yung desired na gene natin. Okay? Ayan. So, ayan, ikakat na po natin siya downward and then the other po is upward or vice versa po. Ayan. So, the fragments now are transferred on the membrane and then ma-hybridize po siya gamit yung probe or yung PR2 or yung fluorescence dye natin followed by ayan, so maging gano'n, followed by auto radiodiography. And then yung autodiography natin, the presence po nung band na yun indicates that the gene is actually present. Whereas kapag wala po nagpakita na band, ibig sabihin absent po yung gene. Okay, so for example po, pansinin po natin tong sample. So we have here the samples. Sa 1, meron pa tayong isa. Sa 2, wala po tayong nakikitang band. So meaning absent po yung gene. And then sa 3 po natin, meron pa tayong nakikitang dalawa. So meaning present po yung gene natin doon. And then for number 4 po, wala din. So meaning absent po yung gene natin. So with the use po of probe or under the autodiography, ganito na po yung mangyayari. Makikita na po natin siya directly. Okay? This actually indicates that the gene is present or absent, yung probe po natin. And by that po, we can also detect a uh, copy a copy of number of genes in this example. So makikita na din po natin kung ilang copies yung meron or ilang copies wala. So dito, let's say this sample 3, meron tayong nakikitang dalawang bond. Yung dalawang bond na yun indicates that there are two copies po ng gene in that certain genome. And then on the other hand po, sa sample 1, meron po tayong nakikitang nag-iisang bond. So whereas, kapag isa lang po yung bond, meaning po, nag-isa lang po yung gene natin in a genome. Okay? So here, how it works in the gene. So present at the second location. Ayan. So again, magsisequence na naman po siya. So the presence na po nung restriction sites nung upstream and downstream natin to the gene will be different. So ang mangyayari na po ngayon is that magdadigest na po siya. And then doon na po makikita natin yung overall length na nung DNA fragment. Okay? Ayan. So using this technique such as RFLP or the restriction fragment length polymorphism, and the other one po is the AFLP or the amplified fragment length polymorphism. Okay? So any questions po with regards to Southern blot? If none, ayan. So I have here a few questions na po. Ayan. So before we end our discussion. So first question, ayan. So what type of labeled probe is used to identify DNA fragments in Southern blotting? Diba? So ayun na yung clue natin dito. So we have the labeled as the complementary DNA probe or yung fluorescence type po natin. Okay? Another question. So which of the following is separated during digital electrophoresis when in terms of Southern blot? A, DNA RNA fragments, B, DNA fragments, C, protein molecules, or D, liquids? The correct answer is DNA fragments and not RNA fragments. Ayan. So, Southern blotting relies on the binding of DNA to complementary RNA probe. True or false? False. Hindi po siya nagre-rely sa binding ng ating DNA to a complementary RNA probe. Ayan. So, sino na po nag-develop ng ating Southern blotting? Southern. Ayan. So, it's Edwin Southern. And then, what is used to visualize the DNA fragment in Southern blotting? We call that as, again, yung fluorescent probes natin or yung PR2. Ayan po. And then, last question. So, the membrane in Southern blotting is used to immobilize DNA fragments for detection. Is it true or false? Ayan. So, the correct answer po here is true. Okay? It's true po. Ayan. So, with that po, that will be the end of my report on the tools for studying nucleic acids. So if you have any questions po, you can PM me or ask questions po about this. Ayan. So thank you so much po for listening and see you po on next meeting. God bless po everyone. Goodbye. And then, so what is the enzyme essential for DNA replication in the PCR? So we have the DNA replication in so the enzyme essential for that is we have the Taq polymerase that we've mentioned. Okay. Okay, so sorry for that.